Germplasm banks. Germplasm banks are the establishments concerned primarily with the conservation of the genetic material which may be lost through genetic erosion. In practice, germplasm is a plant part. It's a plant part from which new plants can be generated. Currently, techniques of preservation have greatly increased the usefulness of germplasm banks by the application of tissue culture and cryopreservation techniques. Most of the plants can be maintained in a viable state for very long periods of time. Cryopreservation is the preservation of germplasm at an ultra low temperature of minus 196 degree, temperature of liquid nitrogen minus 196 degree, minus 196 degrees Celsius. At such a low temperature, biological activities cease to take place, cell division stops, and no genetic change would occur. It is especially important in the conservation of vegetatively propagating crop plants. In this minus 196 degrees Celsius, that is the temperature of liquid nitrogen, all the biological activities of this germplasm will stop and cell division will be stopped. By tissue culture techniques, meristematic cells or small parts of vegetative tissues are allowed to grow in a suitable nutrient medium to form small clumps called calyces. These calyces are then preserved in cooled liquid nitrogen. Cryopreservation. They can remain viable over an unlimited period of time. They can be taken out and allowed to multiply as and when required. Often dried orthodox seeds. Orthodox seeds are seeds which can withstand lower temperature along with lower water content. So with very low moisture content and stored in cooled liquid nitrogen at minus 196 degrees Celsius. These seeds can remain viable for an almost infinite period of time, even for several centuries. Tissue culture method of germplasm conservation is especially significant under the following conditions: when specific genotype have to be saved. When we are saving the genotype, tissue culture technique is very important. When the seed progeny is highly variable, we will be conducting tissue culture to save that germplasm. When the seeds are recalcitrant, what are recalcitrant seeds? Recalcitrant seeds are seeds which can't withstand lower moisture content or lower temperature. They become, dead, or they won't remain viable after that. When seeds are altogether absent, we will be conducting tissue culture and saving the calyces for later use. And after creating the calyces, we will be preserving them by cryopreservation. Tissue culture method of conservation is advantageous in two ways. Facilitates the storage of a large number of genotypes. Here we are going to save the genotypes in a very small area in culture vessels at an extremely low cost. Here cost is very low. Provides an effective means for multiplying endangered species of plants and thereby enables their reintroduction to their original habitats where they have become scarce or lost. We will be reintroducing these plants after tissue culture. Germplasm loss is a major concern in Asia, parts of Africa, Southern Europe, and countries broadening the Mediterranean, where antiquate cultivars are rapidly replaced by new varieties. With the loss of older cultivars, the traits and qualities possessed by them would get permanently lost. To avoid this, such plants can be conserved in gene banks and germplasm banks. The International Board for Plant Genetic Resources, instituted in 1974 with its headquarters in Rome, is now playing a pivotal role in the conservation of endangered plant wild plants and cultivars. Under its initiative, a chain of gene banks and germplasm banks have been set in the different parts of the world. Here they are talking about International Board for Plant Genetic Resources. International Board for Plant Genetic Resources. So this is a mode of ex situ conservation. Germplasm banks.